Everybody's taking at least one.
Like some of us, it happens a little later in the game. But you know what? It makes you realize.
Makes you appreciate. Don't forget to check in on attendance. Check in on attendance, please.
Like the 76 or something. Yours? On like out of all of my chemistries I've ever taken. Yeah. Well, it's a 70 something.
Check in on attendance, please.
Yeah. Zach texted me after the first organic test. He's like, "How do you do an exam?" Because I'm always like, "Oh my God, that was so hard." Like, you know, as I do.
Of course.
She's not wrong. He was like, "How'd you do?" And I was like, "Oh, so check." And I was like, "Yeah, I made a 61." And he was like, "Oh my God, me too."
I think my worst grade ever was physics.
Really?
All right. So why did the baby strawberry cry? Because his parents were in a jam.
Oh my God.
Oh,
That's kind of dark.
That's just sad.
It is kind of dark. It's a little bit dark. It is. Okay, so I'll tell you the one Charles really liked, even though it's terrible. You ready for a really terrible one?
Yes.
Like he laughed and laughed at this, and it's terrible. What is green and bumpy and black and white? And black and white.
That's a lot of things.
A pickle in a tuxedo.
Why? I mean,
I don't know. I don't know. It's saying you're present.
I've never clicked it.
It's saying you're present. Call it good. Just call it good. Saying you're present. All right. So we are starting chapter 11. Yay! You have to get to this point. You have to. Yes, you do. Yes, you do. So what have we made so far? Have we made ATP? Can we do anything in the cell? We don't have ATP yet, right? We made a little bit. We made a little bit, just a tiny bit. We are about to make a ton of ATP. We are about to make a ton of ATP. So all of these energy conversion processes, that's what's like really going to sustain life on Earth. And so this is massively important. We made energy carrying molecules in glycolysis and in citrate, right? What were those energy carrying molecules? NADH and FADH2. Okay, now we're going to figure out how we make ATP from that. And we do that with oxidative phosphorylation. Okay, all right, so in this one, in order to understand how that works, you have to understand how the chemiosmotic theory is designed, because that's what oxidative phosphorylation is based on. Then we're going to talk in specifics about all the proteins and enzymes inside of the electron transport. I call it the electron transport chain. I am too old to change. So I don't call it the system, I call it the chain, whatever. It's always been, but now they're making the move. It's a system, it's not a chain. ETC, electron transport chain. Okay, yeah, okay. So then, right, after the electron transport chain, at the very end of the electron transport chain, the last thing that has to happen is that we have to actually make ATP using the ATP synthase complex. And then we'll talk about important transport systems within the mitochondria, and then how oxidative phosphorylation is regulated. So we'll see how far we get. What we get is what we get. So the chemiosmotic theory was really, really a revolutionary theory. And it's one of the really great unifying principles in chemistry biology, right? But it was insanely radical at the time that it was proposed in only 1961, which like was not that long ago. Really? If you kind of think about it, it's not that long ago. What's that? Yeah, yeah. Just barely, just barely. So really this system is conserved in all organisms, right? That's saying a lot. It's not just conserved in some, it's conserved in all. So what is the chemiosmotic theory? So this is going to happen in the mitochondria. It also happens in photosynthesis in the thylakoids, but if you want to know about that, you got to take biochem 2. We're just going to look at it in the mitochondria. So inside the mitochondria, we are going to create a proton gradient across our inner mitochondrial membrane. So you know how the mitochondria is arranged, right? You have an outer, oh, come on, I hate when I do this. You have an outer mitochondrial membrane, right? This is the cytosol. This is the outer. Then you have a space, and then you have the inner membrane. This is the matrix, right? And this is the inner membrane space. Yes, sort of. Okay, so we're going to establish this gradient across the inner membrane, which means that we're going to pump hydrogen ions from the matrix into the inner membrane. So they're not actually going to leave the mitochondria. They're going to stay inside the mitochondria, but then they'll be in the inner membrane space. Then we're going to use this high concentration of hydrogen ions to basically drive ATP synthase and make ATP. And then the hydrogen ions will come back to the matrix and they'll keep cycling through that process, okay? And so all of this is really gonna depend on diffusion, right? Everything wants to go from areas of high concentration to areas of low concentration. So we're gonna use the energy that's stored in those NADH and FADH2 molecules that's energy, right? To pump hydrogen ions against the gradient. So we're gonna pump them from the matrix to the inner membrane space. Now we've got a ton of them sitting in the inner membrane space. Well, that's potential energy, right? So then I can harness that potential energy of all these hydrogen ions that are sitting in my inner membrane space, and then they will flow through the ATP synthase molecule. And when that happens, that will be the energy that drives ATP synthesis, okay? So you have to remember that it's completely dependent on concentration. Hydrogen ions want to move from areas of high concentration to low concentration, right? And in the end, we're gonna make ATP. So the movement of protons down the concentration gradient, really this is through the ATP synthase complex. Okay, so it's not just that they're diffusing across the membrane. They're actually going through the complex, and that movement of the hydrogen ions through the complex is what is going to result in ATP being produced. Okay, so this same process is used in eukaryotes. It's used in bacteria, very, very, very highly conserved. And so we see it not only in ATP generation in oxidative phosphorylation, but we also see it in the creation of sugars, right, in plants that use photosynthesis. So it's really, really, really cool. So our proton gradient is initiated by an outward pumping of hydrogen ions. So, okay, so let's draw what we're doing here, right? Okay, if this is the matrix, okay, what am I gonna have in the matrix? I have hydrogen ions that are basically sitting around in the matrix. And we are going to use what molecules? NADH and FADH2. These are carrying electrons, right? So those electron carrying molecules are going to transfer those electrons to, and I'm gonna draw it as one big box, and I'm gonna call it the ETC. It's really 4 different complexes, and we'll learn all the enzymes and all that kind of stuff, but let's just draw it as one thing for right now, okay? So simple, and then we'll build on top of it, right? Okay, so what's gonna happen is that eventually, what's going to pick up those electrons? Like, what's the final electron acceptor? Oxygen. So we have to have O2 inside of our matrix. So our NADH and FADH2 are going to give up their electrons. They're going to get picked up and passed around. So remember how we talked about the hot potato thing, right? And we talked about, I know, right? Y'all like, what is she talking about? When we looked at the different electron acceptors, right? And we looked at their ability to accept electrons, right? So what do we call that? The reduction potential, okay? So each substance has a reduction potential value, right? And O2 has the most positive, right? And so our NADH and our FADH2 are kind of down at the bottom in that table. You remember that table? And there are lots of things in between. So what ends up happening is that NADH and FADH2 pass their electrons, and those electrons get passed around like hot potatoes until we get to oxygen. Oxygen is the final electron acceptor. So those electrons get passed around until the very end, where the electrons are picked up by oxygen, right? Now, in that conversion of passing around those electrons, hydrogen ions get pumped through the electron transport chain in multiple places, but we're only going to draw it once, right? So now what do I have on the outside? This is the intermembrane space, right? Inner membrane space. I now have what? A very high concentration of hydrogen ions, which is awesome. Now, those hydrogen ions are going to pass through another thing, and we call this the ATP synthase complex, right? So the first one we did, the proton gradient is initiated by outward pumping. This is the red, outward pumping of hydrogen ions from the matrix by 3 large protein complexes. So that's what we kind of drew in red. Now, the inward flow of hydrogen ions through the membrane-bound ATP synthase accomplishes ATP synthase. So what happens here, if I can extend the page, right, is that our hydrogen ions are going to come out the other end, right, and will be used for subsequent cycling, right? So you got to think about the cell always being very, very eco-friendly and recycling everything. And so this conversion, this pulling our hydrogen ions from areas of, this is high concentration to areas of low concentration, are going to actually drive ATP synthesis. So I'll have ADP plus an inorganic phosphate, and I'll turn that into ATP.
It says ATP synthase complex.
Yep, complex. I tried to fix it. I didn't fix it. Complex. Okay. So what we've basically done here, right, is establish a proton gradient. So the goal here is to establish proton gradient. We converted those energy carrying molecules into a proton gradient. Then we convert that proton gradient into mechanical movement in the ATP synthase complex, which then translates into phosphorylation of ADP, and you make ATP. So it's kind of pretty gosh darn awesome. So what kind of reactions have to happen in order for us to move these electrons along? With redox reactions, right? So we call it oxidative phosphorylation. So the oxidative part is talking really about the electron transport chain and how we're moving those electrons around. What's the phosphorylation part? The ATP synthase. What are we phosphorylating? ADP into ATP, right? It's just an addition of a phosphate group. And so that's why we call it oxidative phosphorylation. They actually named it well.
Where does the O2 go after
it turns into water? It turns into water. So that's why we say water is a waste product, right? Okay. All right, so our electron transport chain, right, really talks about just those redox reactions that occur in a set of protein complexes. And all of these complexes, just like we drew here, are embedded inside of what? The inner membrane, the mitochondrial inner membrane, because, right, we have another membrane like out here, right? So this is the outer membrane. This is the inner membrane. And that's the matrix, right? Okay, so you have to kind of think, you have to keep the structure in mind, right? Okay. So what's going to happen is you're going to have NADH being oxidized to form NAD+. What else is going to happen? You're going to have FADH2 being oxidized to form FAD. And at the end, the very last thing that happens in the electron transport chain is that O2 is reduced to H2O, right? So this is why it requires, oxidative phosphorylation requires oxygen, and that's why we do it in the mitochondria. Question?
Is there a reason that FAD always takes 2 protons?
It's a stepwise thing.
Oh, okay.
It's a stepwise, and whenever you make NADH, you also make a free proton, right? So we just don't show it that way because it's, I don't know, it's whatever, right? So if you look in that table, if you look in your reduction potential table, if we look at, for example, NAD plus NADH, if you go and look up those potential values, what you get is negative 0.32 volts. If I look at O2, and it's really 1/2 O2 and H2O, and we'll talk about why it has to be that way, it's positive 0.82 volts. I thought it was 0.83, but maybe I wrote that down wrong. But anyway, that's a really big difference between the two. So that you're looking at the lowest to the highest within oxidative phosphorylation. That's a really big difference. So do you remember in one of the sheets I gave you that I asked, how do we harness energy in redox reactions? Do you remember that? One of those is that we have to go through multiple small steps, right? And that's exactly what we're going to see. Lots of little small steps. And some of them you'll have to know, and some of them I'll just generally say, this sort of happens. But you're going to make a lot of ATP, and so you're going to have a lot of free energy after this all happens, right? But really, if you kind of think about this, there are 2 things that you have to remember when you are pumping hydrogen ions. There are 2 effects that you're going to have. One is that because it's hydrogen ions, what are you going to change? pH. So you're going to have a change in pH across the membranes, right? The other thing is because those hydrogen ions are ions, they are charged. So what else are you changing? Electrical potential. Electrical potential. So electrical potential has to do with charge, right? Remember, like we talked about neurons, right? And that you can pump potassium and sodium, right? And you can get a differential charge. Same thing happens in the mitochondria. Even though it's positively charged hydrogen going to areas of more positive, you still have an area that is more positive because you're pumping it, and an area that is less positive. So we call it the plus and the minus, even though it's not really a minus. But you know what I mean? Okay. So when you're looking at the 2 sides, right? In here, the yellow area is the matrix, right? And then out here, we're talking about the intermembrane space. So when we talk about this, we talk about the P side and the inside. The P side is the inner membrane space, and that is because it is P4, guess what? Positive. P4 positive, right? So you have more hydrogen ions here. Because the hydrogen ions are going to have to flow down a concentration gradient, you have to have more hydrogen ions on this side of the membrane. So this is your high concentration of ions, right? See, right here, high concentration. On the other side in the matrix, you have a low concentration. So we call this the N side or the negative side. So this is very similar to an electrical circuit, right? So you have a resistor, right? The resistor is whatever performs the work. So what's performing work here? My ATP synthase complex. This is what I want to drive, right? I want to make ATP. I have a capacitor that's going to temporarily store some energy. That's going to be my proton gradient, right? That's potential energy. Proton gradient is potential energy. If I allow those hydrogen ions to flow back through the membrane, I can harness the energy from their movement to drive my ATP synthase complex, right? And then we have the battery, and the battery itself is the electron transport system. So this is a really great system, and it cycles. But sometimes you want to basically uncouple the two. You want to uncouple the electron transport chain from the ATP synthase. So basically, I'm going to show you a kind of a diagram about what happens. But basically, when that happens, you've got that potential energy, but then you allow those hydrogen ions to flow back into the mitochondrial matrix without going through the ATP synthase complex. So you don't make ATP, but what you do make is heat. So this is really important in some organisms, especially ones that hibernate, right? This happens in brown adipose tissue because they have to keep things like their brainstem relatively warm in the winter time. And so you have certain tissues that uncouple this process that then allow leakage, and so you can produce heat. So what I want you to do, I want to show you the animation of how this works. It's a really good little animation. And I'm going to rewind it. I know there's no sound. I know. I'll rewind it.
Energy in eukaryotic cells is generated in the mitochondria through energy transformations in oxidative phosphorylation. Most cellular energy in eukaryotic cells is generated in the mitochondria through the process of oxidative phosphorylation. This process consists of a series of energy transformations that result in ATP synthesis. In mitochondria, ATP synthesis begins when redox energy from the electron transport system is used to move protons across the inner mitochondrial membrane, creating a proton gradient. This proton gradient is a source of electrochemical energy, which is converted into chemical energy as protons move through the ATP synthase complex, generating ATP. Let's take a closer look at each of these energy transformations. The process of oxidative phosphorylation requires the electron transport system, consisting of 4 protein complexes and an ATP synthase complex embedded within the inner mitochondrial membrane, as well as associated electron carriers. The ongoing synthesis of ATP in the mitochondrial matrix also requires 2 transporter proteins that provide inorganic phosphate and ADP for the reaction, while at the same time exporting the newly synthesized ATP. The electron transport system captures energy from a series of sequential redox reactions. Oxidation of NAD.
So look at the difference, right? We had 2 energy carrying molecules. They start out in different places in the electron transport chain. That's really, really, really important. And that will translate to different amounts of ATP being generated from one molecule of NADH versus one molecule of FADH2, right? So NADH comes into the system, the chain, earlier than FADH2, okay?
NADH and FADH2 releases electrons. We will follow electrons from NADH, which enter at complex 1. These reactions are coupled with the movement of protons across the membrane. The electrons flow through the electron transport system. And reduce oxygen to form water.
I swear I didn't touch it. Sorry.
The redox energy released by these reactions is used to translocate protons from the mitochondrial matrix to the intramembrane space, creating a proton gradient. Protons move back into the mitochondrial matrix through the Atp Synthase Complex, generating ATP. This movement of protons across the membrane down the concentration gradient to generate ATP is called chemiosmosis. Directional proton translocation by the electron transport system increases the proton concentration on one side of the membrane, creating a proton gradient. This proton gradient is a source of electrochemical energy due to both the chemical gradient, pH differential, and electrical gradient, charge differential. Chemiosmosis takes advantage of this electrochemical gradient, which causes protons to flow back into the mitochondrial matrix through the Atp Synthase Complex, providing energy to drive ATP synthesis. The electron transport system can then recycle these protons, completing the proton circuit. In plant chloroplasts, chemiosmosis is also used to synthesize ATP. Chloroplasts use sunlight energy rather than redox energy to establish a proton gradient across the thylakoid membrane. The proton circuit behaves like an electrical circuit, as potential energy is converted to work as a result of movement of charge. Proton circuit uncouplers disrupt proton flow by allowing proton leakage back across the membrane without the usual production of ATP. This converts redox energy to heat instead of ATP synthesis. Certain compounds, such as oligomyosin, can inhibit the Atp Synthase Complex, blocking the proton circuit so that oxidative phosphorylation cannot occur, eventually leading to cell death. We can see that each part of these energy transformation processes is vital to sustain life.
Okay, so when they were talking about, right, our uncoupling, right? So this happens, this is the uncoupling event that they were talking about in the video. This happens in brown adipose tissue. Do you want this to happen in all tissues? No, absolutely not, right? Because we need to make ATP. If we don't harness those, that electrochemical gradient of protons, then we're not going to make ATP. But there are some cases where heat is more important than making ATP. So for hibernating animals and things like that, to maintain the right metabolic temperature, that's what you need to do, right? Okay, the other situation she was talking about, that's an uncoupling, is oligomyosin. Anybody know what oligomyosin is? It's a what? Well, no, it's not myosin. It's an antibiotic. So oligomyosin, it's specific for bacterial ATP synthase, right? Even though the complex, the electron transport chain and the ATP synthase complex is conserved throughout all organisms, it does not mean that there are not structural differences and sequence differences between eukaryotic and prokaryotic, right? And so we can take advantage of the fact that prokaryotic ATP synthase will bind oligomyosin, but it will not bind to eukaryotic ATP synthase. So we can cause cell death in prokaryotes, but not eukaryotes, right? So it's very easy to treat, and I say easy, it's relatively easy to treat somebody who has a prokaryotic infection. But when you have people who have a eukaryotic infection, right, it becomes more difficult because whatever drug you give to the person to kill the parasite, to kill whatever it is, will actually be toxic to the host as well, right? Because you're both eukaryotes. So, you know, it's just a good example of how that works. Okay, so let's take a step back and remember our mitochondrial structure. Remember we said that we have an inner membrane and an outer membrane. What's different? What do you notice different about the inner membrane versus the outer membrane? It has, yeah, it has folds in it, almost like the stomach does, right? Wasn't that really cool when we dissected that and looked at the inside? Yeah, that was really cool. All right, so it has lots and lots of folds. So what has more surface area? The inner or the outer? The inner has lots and lots more surface area. So when you talk about mitochondria, right? I'm gonna do a little math for you here. Any given cell has 10 to 100 mitochondria per cell, right? You know, it depends on what cell type and what the energy needs of the cell are, how many mitochondria you're gonna have. You do not have to know these numbers. You do not need to memorize these numbers. This is just a cool thing, okay? So we have 10 to 100 mitochondria per cell. So our surface area, surface area, woo, is 5,000 micrometers squared. Okay, so if we have that, so these give you this surface area. How many cells do we have in our body? More than that, you have about 1 trillion. You have about 1 trillion. Somewhere between 10 to the 13 to 10 to the 14, somewhere in there, you know, trillion. So what does that translate to in surface area? It translates to 185,000 meters squared. Oh my God, I can't write today. Meters, and I'm writing an eraser, meters squared. Oh my God, that's a lot. That's like 26 soccer fields per person. A sport? Soccer? S-O-C-C-E-R? Soccer. Whatever. Oh my God. I told y'all I don't spell. I just write. I'm thinking and writing. All right, so 26 soccer fields per person of surface area. That's a lot. That's a lot. I'm trying to like emphasize that's how much surface area you have, and this is microscopic. So how many ATP synthase complexes do you think you have? Right, a lot. I can't even say a lot enough to emphasize how many that you have, right? So the reason that you are able to make tons of ATP, I mean, your cells use ATP for everything, is because of these folds inside of the inner mitochondrial membrane, which increase their surface area. And so we call those folds the cristae of the mitochondrial. So what's really good about doing this in the fall is that this is right about Thanksgiving, Christmas time, right? So around that time, what does everybody have for dinner? Turkey, right? Okay, now everybody in here I know has a preference, right? Dark meat or white meat. And everybody is, whatever your preference is, if you get served the wrong one, it is a problem, right? Why is some meat dark meat and some meat white meat? More mitochondria. If you're eating dark meat, you like to eat mitochondria. I like mitochondria. I much prefer my meat with excess mitochondria. And I want y'all, like, when the next holiday season comes around, instead of saying, may I have some white meat or dark meat, I want you to say to whoever it is, I'd like mine with the highest mitochondrial content, please.
everybody would look at me like they didn't know I was thinking differently.
Then it sparks the conversation. I mean, that's amazing, right? So if you look at, so how many people in here are hunters? Okay, when you kill a duck or something like that, do you see white and dark meat? Why not? It's a great question, right? It's a great question.
It's also because they use their muscle to run more.
That's what it is. So commercial turkeys don't fly. They don't fly, right? And that's what the breast, the white meat, controls them flapping their wings, right? So they don't really flap their wings. So do they need ATP? No. So are they going to have a lot of mitochondria? No. But wild, do they fly? All the time, right? So you have basically all dark meat. Didn't you always want to know that? They're like, no, not really, but okay. Don't you feel so satisfied at this point? Okay, so now we have to get into some nitty gritty. So these are our protein components of the electron transport chain, right? What's that? Yeah. This is still section 1. So each of the complexes contains a specific enzyme. So they are enzymes, right? Complex 1. What energy carrying molecule comes in at complex 1? NADH. So the enzyme in complex 1 is NADH ubiquinone, which ubiquinone is going to take those electrons from complex 1 to complex 2. So it's NADH ubiquinone oxioreductase. What kind of reactions are happening? Redox reactions, oxidation reduction reactions, oxioreductase. Yay, they did something well. Yes. Yes, we're going to talk about it in detail. Don't worry. Don't worry. Okay, complex 2, succinate dehydrogenase. Where have you heard that before? In the citrate cycle. It's actually a citrate cycle enzyme, but it also functions in the electron transport chain. So what comes in at complex 2? FADH2, okay? All right, complex 3 is ubiquinone, because the ubiquinone is taking it from complex 1 to complex 3. So then, in order to go from complex 3 to complex 4, we have to go through cytochrome. So ubiquinone and cytochrome are carriers within the electron transport chain. So we go ubiquinone to cytochrome C within complex 3, and it's again an oxioreductase, right? And then the very last one, you end up with cytochrome C. That's our last carrier molecule, bringing it to complex 4. So we call that cytochrome oxidase. So the names actually kind of make sense, except for succinate dehydrogenase, but that's because it's in the citrate cycle. So it was named first in the citrate cycle. Then it was discovered that it was in the electron transport chain, so it carries the name from the citrate cycle.
So did succinate dehydrogenase, just like I like to make sure I'm thinking about it right, in the citric acid cycle, it does the same thing with FADH2 or no?
Yeah, we're in, yes,
Okay.
yes, yes, it's FADH2, yes. So all of these complexes that we talked about so far, they're embedded inside of the inner mitochondrial membrane. So they have parts that face both the inner mitochondrial, well, you have some that are, they're all embedded in the inner mitochondrial membrane, but you have transmembrane domains that function so that they can communicate from one complex to the other complex. And then you also have some that face down into the mitochondrial matrix, because where does citrate cycle occur? Remember, we do glycolysis in the cytosol. What's our product of glycolysis? Pyruvate. What do we do with pyruvate? Yeah, but what do we also have to do with it? we have to bring it into the matrix. So where does citrate cycle occur? In the matrix. And so that's why complex 2 has a functional domain that faces into the matrix, because it has to work in the citrate cycle and it has to work in the electron transport chain. Yeah? Okay. All right. So this is our more complex drawing of oxidative phosphorylation, right? Okay, so we have NADH that's going to come in at complex 1, right? The NADH gets converted into NAD+. It drops off its 2 electrons. As those 2 electrons get passed, and they do this as this green arrow, but I kind of like to do it as kind of a zigzag because they get dropped off with different molecules within complex 1, because complex 1 is not just one enzyme, right? But it's composed of things with different reduction potentials so that we can move those electrons along. So then it's going to get picked up by, what was our molecule? Ubiquinone. Ubiquinone, abbreviated by the Q, right? Is going to pick it up here at complex 1, and it's going to bring it over to complex 3. The alternative is that FADH could come in at complex 2, right? And then those electrons will go through complex 2. But look at complex 2. Is complex 2 moving any protons? No. Is complex 1 moving protons? Absolutely. Complex 1 moves 4 protons, right? So our electrons move from either 1 or 2 into 3. When we move into 3, what happens? What do we pump? More protons into the inner membrane space, right? So if you look at 1 and 3 so far are pumping protons. Then those electrons move 1 at a time. In ubiquinone, they move 2 at a time. In cytochrome C, they move 1 at a time. That's why we say 2 times 1 electron, because they're moving really only 1 at a time at that point. Then they enter into complex 4. When you're in complex 4, that movement of electrons will now only pump 2 hydrogen ions out of the matrix into the inner membrane space, right? So when we get to complex 4, this is where we will convert our oxygen into water, right? Because that's our final electron acceptor. Okay, so if I come in with NADH, how many protons do I pump? 4 here, 4 here, 2 here, right? So how many do I pump? 10. If I come in with FADH2, how many do I pump? 4 here, 2 here, 6. It's a hard math day. It always is. So in order to drive the phosphorylation of 1 ADP into 1 ATP, I need to move 3 hydrogen ions down the concentration gradient from the inner mitochondrial space to the matrix. So I'm going to require more FADH2 to make that happen than I will NADH, right? Just because of where they enter into the complex. So the other interesting thing that you have to keep in mind is that I need, in order to phosphorylate ADP, I need to get ADP into the matrix, and that's not where it normally resides. So I actually have to have some kind of a transmembrane protein that will facilitate that translocation. So every time I pump out a made ATP, I pump in an ADP. So what do we call that kind of a, well, it is a translocase, but it's a symporter and antiporter. Antiporter. One goes in, the other goes out. Antiporter. This one, for every 1 hydrogen ion, because not all the hydrogen ions are going to be used by ATP synthase, I also need to be able to pump in inorganic phosphates. So for every 1 hydrogen ion, I can pump 1 inorganic phosphate into the matrix. So what kind of a pump is this? Symporter. Okay, so now I have all of the components that I need to make the electron transport chain and ATP synthase go, okay? All right, so what I wanna do, what time is it? 34. Okay, we can't answer everything, but I want to try, we can answer most of this. We can answer most of it. I want y'all to try. You're probably gonna need your notes for at least number 3. And follow them first. It's not that big. It's not as bad. And I didn't make myself one of these. So I'm just going to go and extend a page and do that. I believe it is 28. It is 28 today.
Yeah,
2 days. I know Friday.
Surely gonna move down that.
Are we doing this together or y'all want to try?
No, I want to just go.
Y'all just want to do it together. Okay. All right. So what does oxidative phosphorylation accomplish for us? What do we get? We make ATP. We make ATP. That is the name of the game. Make ATP. So we're going to make ATP. We're going to make 28 ATP. We're going to make 28. How many have we made so far? In citrate, in glycolysis? 4. We've made 4 so far. So 28 of the 32, right? We can kind of do this. 28 of the 32. So most of the ATP that's made in the cell, is it made in glycolysis? Is it made in citrate? No, it's really made in oxidative phosphorylation, right? So it makes ATP, but from what? How do we get it? That's true. So how do we get an ADH? From the oxidation of, we've talked about so far only sugars, right? But what happens if you go on a low carb diet? Through you break down fats, you break down triacylglycerides. So oxidation of, we'll say metabolic fuel. And we'll put in parentheses sugars, and we'll put fats, right? Okay, so we're going to make ATP from the oxidation of metabolic fuel. So we oxidize that metabolic fuel, and then we do, like, there's 2 steps. One's the oxidation, and the other is the phosphorylation. So from the oxidation of metabolic fuels to perform substrate level phosphorylation. What is substrate level phosphorylation? This is AKA make ATP. Okay. All right, next one is to write our equation for this. So let's write this equation just for NADH. So we need NADH.
Are we gonna have to, like, write the equations and everything?
Possible.
It's probably like that.
All right, what else do you need? If you want to make a proton gradient, what do you need? Protons, we need some protons. We'll balance it in a minute. and I want to get out of this ATP. So what do I need to put into it? A DP and PI. Plus, we need O2. Okay, so those are our substrates. What are we going to make? Well, let's start with what we have, right? If we had NADH, what are we going to have at the end? NAD plus. Now, what are you going to do with the hydrogen ion? We're going to make H2O. Okay. Then we're going to make ATP. Anybody want to attempt to balance? It's not a half. It's not a half. Look at your waters. You need 2 waters. So you need 2 hydrogen ions. Right? You're going to have to put a 2 in front of what? 2 in front of the NADH. So 2 in front of the NAD plus.
Why would I try it? Because you have 4 hydrogens. Oh.
It's just balancing. It's okay.
Yeah.
It's okay. It's not that big a deal. And then you end up making 5 ATP, 5 ATP, 5 PI. Y'all got close? Not bad. Not bad. All right. What are our key enzymes? Key enzymes. So remember the first one. What's coming in at the beginning? What's coming in complex 1? NADH comes in, and it's going to be picked up by ubiquinone. So NADH, ubiquinone, UBIQUINONE.
Okay, I'm going to spell that, but not soccer.
I wasn't trying to spell soccer,
Okay.
but I probably would get it wrong if I tried anyway. Oxio, OXIDO, REDUCTASE, right? Okay, so well, right. So what is this? NADH, ubiquinone, oxioreductase. This is really complex 1. Complex 1. So this is our very first oxidation reaction where we have NADH oxidation coupled to our NADH releasing its electrons, right? You're like, okay. All right, what's next? So complex 2. Complex 2 is the one that comes from the TCA cycle or citrate cycle. So this is okay, then what? Complex 3. Ubiquinone, what? So remember, which ones out of these are pumping hydrogen ions? So 1 is pumping 4 hydrogen ions. Is 2 pumping anything? No. I wrote 2, 3. 3 is pumping 4 hydrogen ions. Okay, extend your page again and again and again, right? All right, complex 4 is cytochrome C oxidase. This is pumping 2 hydrogen ions. Then you have what was originally called complex 5, but it's not really complex 5. It's not really, is our ATP synthase complex. It's called the ATP synthase complex. I mean, you could call it complex 5, but not many people do that, right?
The ones that just pump electrons are 1, 2.
That just move electrons. They all move electrons. They all move electrons. They all move electrons.
But does it start with complex 1 and
Complex 1 and complex 2 start. So this is where FADH2 will come in. This is where NADH will come in, right? Okay, and then the very last thing is what's an example of oxidative phosphorylation in everyday chemistry, right? And like we talked about using the uncouplers and things like that. So I have another example. It's kind of morbid, but so in Nazi Germany, in the concentration camps, right, they had the gas chambers where they would kill everyone. So they used cyanide gas in those chambers. So what does cyanide gas do? Well, it reduces ATP. It stops ATP production. Why does it stop ATP production? It binds to the cytochrome C, well, the cytochrome, one of the cytochromes. It binds to the cytochromes and it prevents them from being able to move electrons. So you shut down the electron transport system. If you shut down the electron transport system, you don't make ATP and you don't survive. So it's very important to life, right? That's the point I was trying to get across. But you don't have the electron transport chain, that's a very quick death. Very sad, but anyway, on that great note, exams next time I see you. Wasn't planning on ending class on that note, but go enjoy your turkey. Go enjoy some turkey. Please tell someone you know. You know, like when you go to Popeyes, tell them I'd like the high mitochondrial content, please, and see what they say.
It would spit in my food if I said that. I'd be like, this nerd.
So
Sure.
the first one, it's like you're starting off with 32. Is it because you're doing citrate cycle twice because you had 2?
Yeah, we're going to track that.
Okay.
Like, don't focus on the number yet. What I just want you to know is the majority of ATP is coming from the electron transport chain ATP synthase.
Okay.
So from oxidative phosphorylation. We are going to account for every single ATP.
Okay. So just know now that it's 32 total, 28 comes from that. We'll go into the specifics later. Okay.
Yeah.
Second thing, the electrons.
Okay.
I understand like the electrons are really important, but like, do those drive the hydrogens moving across? Like, what is the purpose of the electrons moving?
The purpose of the electrons moving is that they contain potential energy. Whoever's carrying them has a certain potential energy. That's your reduction potential, right? So think about when we say reduction potential, think about potential energy, right? Somebody holding a boulder at the very top of a hill has lots of potential energy. And if we transfer that boulder to people who are standing at different places lower on the hill, that potential energy gets lower, right? And so we use that movement of the electrons, like the movement of the boulder, to drive the movement of hydrogen ions from the matrix into the intermitochondrial space.
Okay, that explains it. Because whenever I was looking at it, I'm like, okay, yeah, I see that the electrons are moving from here to there, but I'm like,
which causes
what's the whole point of them having
this to move here? The whole point is to make a proton gradient.
Okay.
As in, if we can here, the whole point of here is to make a proton gradient. The whole point of here is to use the proton gradient.
So the whole reason for the electrons is that whenever, as it's building up that potential energy, it's making the proton gradient because it's shipping the hydrogens to the
We're transferring the energy from the energy carrying molecules, right, to the hydrogen ions moving, so that they have to go down to a less energetic potential, less energetic, less energetic, less energetic, less energetic, until we get to oxygen.
That's how you move them over so that you get to the ATP.
Okay.
Yeah.
So how you're saying that the gas or whatever the silent gas can block cytochrome.
Cytochrome.
So because it blocks cytochrome, it can't.
Because you basically stop it here, right?
So because of that, well, I mean, like, why would it stop it overall? You're still making it gradient.
Because
you're not. It's a chain. If you have a sequence of events that has to happen, right? You have the guy at the top of the hill with the boulder, right? He's got to transfer this boulder to the next person.
I understand.
I know, but wait. Stick with me. He transfers it to the next one. He transfers it to the next one, right? Okay. What if the fifth person down, he breaks his arms?
He can't transfer.
He can't transfer. What happens to the boulders that are coming in?
They can't be moved.
They can't be moved. So if the boulders can't be moved, you're not translocating hydrogen ions anymore. So you had a proton gradient. You had one. And then you stopped, right? And all of those hydrogen ions went through ATP synthase. They made ATP. There's no more. Then ATP synthase stops.
So you have to get, so like how you're saying, like that 10 hydrogens that are moved outside. So you have to have all of those 10 hydrogens to have that sufficient, like, proton gradient.
You have to have 10 move through, and another 10, 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 another 10, another 10.
It's kind of like a cycle.
No, I get that, but like what I'm saying is like for example, like just from here, that's cut off. You have 8 hydrogens in the inner membrane space. That's not a big enough.
But if you don't have a final electron acceptor, right, you can't transfer anymore.
So that's where that oxygen comes. So
yeah.
Okay. So
because the system is clogged.
That's when the oxygen comes in.
And the oxygen, if you never reduce your oxygen to water, you stop the chain.
Gotcha. Okay, that makes more sense, because like whenever I'm just looking at it, I'm just like, well, you're still making a gradient, but the very last one you have to have oxygen, and the only way that that's gonna
for me to take
reduce the
only way for me in complex 1 to pick up 2 electrons is if I was able to move those 2 electrons to complex 3. The only way complex 3 can transfer to complex 4, right? The only way that each sequential thing can happen if the next one in front of them does their job. Well, if we shut down the activity of complex 4, all the other complexes can't function.
Gotcha. Okay, I understand.
Yeah, it's cool.
Work together.
What's that?
No, they all got to work together.
They do. They do. Oh, and I'm still on Zoom. Who else is participating still? Okay. Y'all have any questions? Y'all stayed on for our awesome discussion. Let's see. I don't think I put up the chat. No, I think they're good. Okay. I will end then. More end.